Hi everyone, welcome back to my new video. My name is Yash Agarwal. I am a second year MBBS student in MDNC Medical College, New Delhi. In this video, I'll be talking about mutation, causes of mutation, and types of mutation. These topics are very much important for a NEET as well as your board examination. So, without wasting time, let's move forward. So, now let's talk about mutation. Mutation is alteration in gene or the chromosome. What is mutation? Mutation is alteration in gene or the chromosome. We know that the genes are located on the chromosome. Let this be the gene. Now, what is the function of a gene? Gene codes for protein, right? Gene codes for protein, and this protein produces physiological changes in the body, right? Now, what if this gene is mutated? If this gene is mutated, it will form abnormal protein or it will form no protein. In both case scenario, the physiology of the body is disturbed. In both the scenario, physiology of the body is disturbed. So, what is mutation? Mutation is alteration in gene or the chromosome. Now, talking about the causes of mutation. Now, there are certain physical or chemical agents which can cause mutation. These physical or chemical agents are collectively termed as carcinogens. These collectively are called carcinogens or mutagens. Now, example of physical mutagens include UV radiation, X-rays, gamma rays, and all the ionizing radiation. Now, chemicals include benzoapyrene. Can you tell where this substance is found? This is found in cigarettes. So this substance causes mutation and thus can cause lung cancer. Similarly, if we talk about UV radiation, how does it cause mutation? It forms thymidine dimers. We know that DNA has thymine, so it will result in forming thymidine dimers. Now let's talk about types of mutation. There are namely 4 types of mutation: point mutation, chromosomal mutation, nucleotide repeat mutation, and fourth one is frame shift mutation. We'll study each one of them one by one. Now, point mutation. What is the meaning of point mutation? In point mutation, the single nitrogenous base pair is substituted by another nitrogenous base pair. So, point mutation, it's a type of substitution, right? One base pair is substituted by another base pair. It can result in 3 case scenario. Either the mutation will be silent mutation, or it can be missense mutation, or it can be nonsense mutation. Let's understand one by one. In silent mutation, the codon remains, the codon changes, but that codon also codes for the same amino acid. For example, if I replace adenine, like you see codes for serine, right? If I replace A with uracil, then also it will code for serine. So this is example of silent mutation. This is an example of silent mutation. But what if I replace cytosine with adenine? It will convert into a termination codon. So the protein chain will not be completed. It will form truncated protein, that is shortened protein. So this type of mutation is known as nonsense mutation. This type of mutation is known as nonsense mutation. Now, if what if I replace uracil with cytosine? It will form CCA. It codes for proline. Now the codon is also changed and the amino acid also changes. So this type of mutation is known as missense mutation. Now can you tell me an example of missense mutation? The example of missense mutation is sickle cell anemia. What happens in sickle cell anemia? In the beta globulin chain, at the 6th position, what happened? Glutamic acid is replaced by valine. Now, why this is important? Glutamic acid is a polar amino acid, whereas valine is a non-polar amino acid. So the globulin chain that is formed under the oxidation stress, it will produce changes in the membrane of RBC. Thus, these RBC will form a sickle shaped form. Now let's talk about another type of mutation, that is chromosomal mutation. In this type of mutation, one gene is not affected, but entire segment of chromosome is affected, which may include many genes. There are 4 mechanisms by which chromosomal mutation occurs: inversion, deletion, duplication, and translocation. What happens in inversion? Suppose this is your chromosome, and this is one segment of DNA, and this is another segment of DNA. What will happen that this segment of DNA will shift? Then what will happen? The blue one is down and the pink one is up. So there is a chromosomal mutation. This type of mutation is seen in a syndrome known as Opitz-Cavalier syndrome. You just need to remember the name. No one asks you on which chromosome it occurs. Now another type of mutation, deletion. In this, what happens? And a segment of DNA is lost. For example, this is a segment of DNA. What will happen to it? It will get lost. So we will have incomplete chromosome. Right? Now example of this is very much important. Cry-du-chat syndrome. In this syndrome, there is deletion of short arm of chromosome number 5. There is deletion of short arm of chromosome number 5. Right? Now moving forward. What happens in duplication? As the name suggests, a segment of DNA is duplicated. For example, the blue one has now the multiple copy of itself. It's also a type of mutation. Where does it occur? It occurs in some cancers, right? Translocation, and this is very much important. What happens in translocation? Now, suppose these are two chromosomes. We have this segment of DNA, and this is another segment of DNA. Now, what was happening in insertion? The segment of DNA was changing on the same chromosome, but here it changes on different chromosome. It is also known as illegitimate crossing over. Illegitimate crossing over. Right? Example of translocation are very much important. Its example is Burkitt's lymphoma and CML, chronic myeloid leukemia. Chronic myeloid leukemia. These are the two examples of translocation. So let's now understand what happens in Burkitt's lymphoma. In Burkitt's lymphoma, we have two chromosomes, chromosome number 8 and chromosome number 14. The chromosome number is important. So on chromosome number 8, we have one gene known as MYC gene. So this MYC gene is translocated to chromosome number 14, which causes Burkitt's lymphoma. So in Burkitt's lymphoma, there is translocation between 8th and 14th chromosome of which gene? MYC gene. So third type of mutation is copy number variation. An example of it is trinucleotide repeat expansion. What is the meaning of trinucleotide? 3 base pairs. Repeat is repetition. So 3 base pairs are repeated again and again. What will happen if the same base pair are repeated? It will result in insertion of many residues of same amino acid. For example, if there is a codon that codes for serine, then if it is repeated, then many copies of serine will be put in the protein, right? Example of this is Huntington protein. What happens in Huntington protein? It's a neurodegenerative disorder. It's a neurodegenerative disorder. That is, neurons degenerate in this syndrome. What happens here? Here, CAG, which codes for glutamine, is repeated on exon number 1, which result in the formation of abnormal protein, which causes Huntington disease. Now let's talk about last type of mutation, that is frame shift mutation. It is very important because many NEET questions are made from this mutation, as well as it can be asked directly in the board. What happens in frame shift mutation? Here there is either deletion or there is insertion of base pair. Either there is deletion or insertion of base pair. In both the case scenario, what will happen? the reading sequence of codon will be altered. And if the reading sequence of the codon is altered, either truncated, that is, incomplete protein is formed, or there is abnormal protein formed. There is abnormal protein formed. One of the most important examples of frame shift mutation, and that too of deletion, is cystic fibrosis. Cystic fibrosis. What happens in cystic fibrosis is that there is deletion of 3 nucleotides. There is deletion of 3 nucleotides, which result in loss of phenylalanine. Which loss? Phenylalanine. At which position? 508th position of CF, transmembrane conductance regulator, CFTR. What is the full form? The full form is CF, transmembrane conductance regulator. CF, transmembrane conductance regulator. This is a channel, this is a chloride channel, and if there is loss of phenylalanine, this chloride channel will be disturbed, and thus it cannot conduct chloride, and it will cause certain symptoms. So here we complete our mutations. So concluding, so mutations can be of many types, but we have 4 broad categories: point mutation, chromosomal mutation, copy number variation, and frame shift mutation. Thanks for watching the video. If you like the video, don't forget to click on the thumbs up button, share and subscribe the channel.